In this topic, we're going to discuss immunity, defense against disease, and phagocytosis. So we're going to look at what is the body's defense system, what is immunity, the definitions for antigen, antibody, and immune response, cells of the immune system, focusing on phagocytes and phagocytosis. So what is immunity? Sir Macfarlane Burnet defined immunity as the capacity to recognize the intrusion of material that is foreign to the body and to mobilize cells and cell products to help remove that particular sort of foreign material with greater speed and effectiveness. The first line of defense is the physical barrier, and this takes a variety of different forms. You've got a protective covering, which is your skin, and this covers the body surface, providing a physical barrier that most pathogens find hard to penetrate. Then there's the epithelia covered with mucus. Now, the body needs to obtain certain substances by diffusion, for example, oxygen and digested food. So you've got parts of the body which cannot be covered by a thick layer like the skin. So for example, exchange regions such as the alveoli and the lungs and the lining of the gut are covered only by a thin epithelial layer. While this still acts as a barrier to pathogens, it is more easily crossed than the skin. Many epithelial cells produce mucus, which acts as a further defense against invasion. Now, in the lungs, the pathogens stick to this mucus, which can then be transported away out of the lungs by the cilia. Hydrochloric acid in the stomach prevents pathogens from going further into the system because it provides such a low pH that the enzymes of most pathogens are denatured, and therefore the organisms are killed. Despite these various precautions, pathogens will still gain entry, and therefore the body has a second line of defense, a series of specific cellular and chemical defenses designed to neutralize any toxins produced by the pathogens, prevent the pathogen multiplying, and to kill the pathogen, for example by phagocytosis, as well as to remove any remains of the pathogen. So examples of chemical would be antibodies, and cellular would be your phagocytes. Let's have a look at a few definitions that you need to be familiar with. The first is an antigen. This is any molecule or organism that is recognized as foreign, and it's usually a protein that makes up the cell surface membrane of invading cells, for example, pathogens. It will initiate antibody formation. So here you can see an antibody, which is a protein called an immunoglobulin. This is made by lymphocytes in response to this antigen. So here you can see the antibody in orange is attaching to an antigen on the pathogen's cell surface. An immune response is when the lymphocytes respond to the presence of the antigen by producing antibodies. Let's briefly look at the cells of the immune system. You've got the phagocytes, which include the neutrophils and macrophages. And then your lymphocytes that include the B lymphocytes and T lymphocytes, or B and T cells. So in this topic, we're just going to look at the phagocytes, and then we're going to look at the lymphocytes in another topic. So if you have a look at this image, you can see the different white blood cells. Notice how much bigger the phagocytes are compared to the lymphocyte. Also have a look at the nuclei. Phagocytes have got a lobed nucleus, whilst lymphocytes have that rounder nucleus that takes up most of the cell. Phagocytes are produced throughout life by the bone marrow. They're also stored in the bone marrow, and their properties are that they are granulocytes and they've got a lobed nucleus. You've got two types, neutrophils and macrophages. Phagocytes remove dead cells and pathogens by phagocytosis and digestion, so they are scavengers. Right, neutrophils move to the blood and they travel through the body. They move out of the blood by squeezing between the capillary cells into the tissue. So here you can see a neutrophil squeezing between those cells. They are released in large numbers during an infection, and they're short-lived, so they accumulate as pus. Macrophages are larger than neutrophils, and they will initiate an immune response by cutting up a pathogen to expose its antigens. They are long-lived. So macrophages are made in the bone marrow. They leave the bone marrow as monocytes, and then they travel to the organ where they mature into macrophages. So they're found in organs rather than the blood. So here you can see some lovely photos of phagocytes. Notice the lobed nuclei in the top three photos. And then in the bottom photos, you can see bacteria being engulfed by phagocytosis. Right, let's discuss the process of phagocytosis. Phagocytosis is the process by which large particles are taken up by the cells in the form of vesicles, formed from the cell surface membrane. In the blood, you've got two types of white blood cells that carry out phagocytosis. Can you remember them? There's the monocytes and neutrophils. So let's go through the different stages of phagocytosis. The first is antibodies attach themselves to antigens on the surface of the bacterium. So the antibodies would have come from the B lymphocytes. Proteins found in the plasma attach themselves to the antibodies. As a result of a series of reactions, the surface of the bacterium becomes coated with proteins called opsonins. Complement proteins and any chemical products of the bacterium act as attractants, causing neutrophils to move towards the bacterium. The neutrophils are also attracted by the histamine that the body cells produce when a pathogen invades them. Neutrophils attach themselves to the opsonins on the surface of the bacterium. They then engulf the bacteria. As they do this, they form pseudopodia around the bacteria. So these pseudopodia are kind of like arms extending around the bacterium. The bacterium forms a vesicle inside the neutrophil called a phagosome. Lysosomes fuse with the phagosome and release enzymes that break down the bacterium into smaller soluble material. The soluble products from the breakdown of the bacterium are absorbed into the cytoplasm of the neutrophils. Okay, here's what I've just said in words. So the antibodies attach to the antigens, the proteins attach to the antibodies, and the surface of the bacterium becomes coated with opsonins. Neutrophils are attracted by the chemical signals and histamine. Neutrophils attach to opsonins on the surface of the bacterium. Neutrophils engulf the bacteria, so keywords to remember are they form pseudopodia around the bacteria. The bacterium forms a vesicle inside the neutrophil, and this is called a phagosome. Lysosomes fuse with the phagosome and release enzymes. And the soluble products of the digested phagosome are absorbed into the cytoplasm of the neutrophils. Okay, so what have we looked at in this topic? We discussed the body's defense system. So we looked at the physical, chemical, and cellular. We looked at what is immunity, the definitions for antigen, antibody, and immune response, the cells of the immune system. So we discussed in detail phagocytes. I mentioned lymphocytes. We looked at the features of phagocytes, where they're made, and then we described the process of phagocytosis. So keywords to remember are antigen, opsonin, pseudopodia, phagosome, and lysosome. And that concludes our lesson. The end.